to Nautilus, and our onshore scientists should be logging back in here
Okay,
in just
great.
a minute. There, they're back.
Yay.
Sorry about that.
I don't think I did anything.
No. Glitch in the satellite. It
Okay.
happens with
Got it.
some
Okay,
regularity,
thank you.
but it's very intermittent. But the system will log you back on as it just
Perfect.
Did. So
and
sorry
at
about that.
least V-Link let me know. No, don't worry. So I didn't just keep talking.
It's all right.
It was nice. Great. Well, the initiative that we're working on the West Coast is starting to wrap up. We've done most of our major field work, and this is a great this whole cruise down the coast that Ocean Exploration Trust is doing is pretty much the last cruise that'll be a big part of the initiative that we'll be analyzing data from. And so the Pacific Fishery Management Council has requested information on, like I said, what's in these areas that have been protected, what's in the areas that haven't been protected. A really large percentage of coastline along the West Coast is protected from some sort of bottom contact fishing. So that happened this year. We added about 40% of the coastline and deep sea water to that protection. So that's pretty exciting. So we're trying to figure out what's in those waters, what's on that seafloor. Most of it is not known yet, as Erica mentioned, especially in the Olympic Coast Sanctuary. So this cruise and this dive in particular are part of something much bigger, and so that's exciting.
Yeah, it's pretty fun how that moves around to different regions, but yet everybody's doing the same sort of research. And when it's not in your region, you can compare what the other guys are doing and maybe learn new tools for the next time that it comes back to where you are.
Absolutely, and it's also fun to see within region learning and expansion, like working on a species guide right now that's not just across California, Oregon, and Washington waters, but also reaching into British Columbia and Alaskan waters, combining our field work along the West Coast and in Alaska and involving the Canadians to get a handle on what kind of species do we have in common and where are the breaks, where do things change. So that's exciting to see too.
Right, because so many species don't recognize state and international borders.
I certainly wouldn't if I were living in the ocean. So a little bit about Olympic Coast National Marine Sanctuary. It was established in 1994, and I wish I could make this a trivia game and ask our audience to answer questions about this and see what people think, but it's a little hard on telepresence. Maybe we'll get there one day, but for now, I just have to tell you. It encompasses about 3,200 square miles of wilderness coastline and coastal ocean off Washington State. And that's linearly about 25 to 50 miles seaward. So the Olympic Coast National Marine Sanctuary is a pretty big area of ocean and of seafloor. It really covers a lot of the continental shelf and several major submarine canyons. It protects a productive upwelling zone and is home to, I think it was 29 species of marine mammals and lots of seabirds too. And everybody on the ship, have you been seeing any of these mammals and seabirds, or has it been too stormy?
Well, we've had a fair number of birds. Yeah, we've had a flock of them sitting around the ship the past few days. I haven't seen too many marine mammals this time. When we were here in 2017, I do remember seeing a couple of whales, but not this time.
Oh, the cruise isn't over yet.
Yep, that's right. Yeah, we had some unexpected finds yesterday in this cold seep we were exploring in Gray's Canyon. We found some pretty amazing seep fauna, but also some pretty cool corals in the periphery, and large fish for that matter.
Excellent. What were some of the big fish that you guys saw?
There were a few sole. I think there was a large one at the end. I think it was a short spine. I'm not familiar with the fishes of the West Coast very much. But yeah, a bunch of large sole, and then seems like thorny heads are everywhere at that depth, around 1,000 meters and shallower. So they're probably the most numerous.
I did see both long spine and short spine
Yep.
yesterday, and to end the dive, we had a really large short spine just hanging out, and they get quite large, up to 40 plus centimeters, and the commercial fishermen call them bucket heads because they have these large, flattened, almost plated, armored heads that are very, very spiny.
Buckethead. So is that kind of an insult, or is that just what it is?
I'm not sure.
I know in the Gulf of Mexico, there's the headless chicken monster sea cucumber that I think that can't be a compliment. One of my favorites. I always post pictures of that around the office when we're actually in the office around Halloween time. Headless chicken monster, I think, is my favorite sea creature.
And that's just
What's your favorite, Eric?
a cucumber that looks like I'm going to have to Google that now.
Oh yeah, definitely. I can put it in the science chat log. It's there's a pretty spectacular video or two featuring that lovely animal.
Enimpniastes is the genus name on that one. Yeah, I haven't seen too many on the West Coast, but definitely Gulf of Mexico and Caribbean, they're quite abundant.
Perfect for Halloween time. Do you have any favorites,
So, Steve,
Erica?
were you a part? I was just going to ask Steve if he was a part of the sample workup yesterday, and if he had any other insights into those two black corals that we collected.
Yeah, I do. Yeah, so we were, we had a pretty marathon sampling yesterday. I think the core team who was sectioning the cores took about 8 hours to run everything.
Ooh.
Yeah, it was a rough one, but you know they got some good data from what I understand. But the corals, yeah, we have one, the white black coral that we were chatting about yesterday. I wasn't on watch when they sampled it, but it does look like chrysopathes. And then there was an orange one, which is more reminiscent of lilipathes. But we're fortunate we got whole colonies of both of those, very small whole colonies. So hopefully it'll be useful to determine species level IDs when they get to the shore.
Excellent. Yeah, they were growing right next to each other, and we were able to grab them both from the same position with the ROV, and the question was, which one is chrysopathes?
Yeah.
When you had them next to each other, and you're like, oh, well, I thought it was this one, but now I think it's the other one.
I'm pretty sure it was the white one was the chrysopathes, but I'd be hard pressed to go to a finer level ID than that on the black corals.
Oh, fair enough. That's very helpful, and they were both quite abundant in that seep area. So Heather, you were asking about my favorite deep sea creatures, or
Yeah, the viewers want to know, or listeners, or both.
viewers want to know. I like anything with big gnarly teeth and glowing eyes.
Also Halloween themed.
Viperfish. Yeah, viperfish, dragonfish.
There's just a hatchetfish in the
Anoplogaster.
camera.
Oh,
Does that count? Hatchetfish in in that category?
hatchetfish are pretty
Oh,
cool.
sorry, in the
have,
Argus camera. It just was out of view.
Oh.
It sounds
They
more
have,
threatening than it looks. I'm a big
Some of
fan
them
of the
are
BC
actually
ones
have
too.
they have see-through abdomens or bellies. You can actually seed into the fish.
That seems useful
We
for
have been seeing
taxonomic
lots of,
ID.
right? We have been seeing lots of myctophids on our descent here.
There was this big
There was.
scattering layer that was tracking our lights, and I think I'm hard with the visual ID, but I think it was cyclophonae, like the other type of myctophid. I mean, it's not a myctophid, it's a gonosomatid, but they're the ones that are thought to be the most abundant, you know,
Right.
biomass in the world. But
What are the bristlemouth?
yes, yeah.
Is that, yeah,
So
okay.
I think it's gonostomatidae is the family, and then this genus cyclophonae is like the one that's just ubiquitous and abundant.
Right.
It's all very hand wavy ID though.
We trust you. You've seen your share,
So
I'm sure.
Steve, what's on your favorite animal list? Is it the hatchetfish, or was that just opportunistically put in by?
Yeah, I'm not sure.
I'm gonna put you down for hatchetfish for now. Change your answer later.
I didn't hear the second part of
Like to
that
look.
question. Can you repeat it?
Just curious about your favorite species.
Yeah, it's always a battle, right?
Coral.
I mean, yeah, I mean that's
Cool.
that's my default, but there's so many.
Yeah, what does your heart say?
Actually, I like sea cucumbers. You know,
Yeah,
deep dark
Somebody
secret.
should.
they are so weird.
Somebody should. That's awesome. Own up to it. Be the one person who loves sea cucumbers.
I think there are more than one, but you know, it'd be difficult to find them all in one room. No, there's something really fascinating about their lifestyle and life history. You know, living in the deepest depths of the ocean, surviving on just like the minuscule scraps of carbon that they can scrounge from the seafloor. And then you've got really cool representatives like the Pelagothuria that we saw a ton last year. These are the midwater sea cucumbers that spend their entire life in the water column. They don't need the benthos, to the best of our knowledge. So they're a pretty diverse, interesting group.
I remember when I first
They
saw the
they're
Pelagothuria, I was like, oh man, the deep sea, I really got to relearn everything I knew. I would not have expected that.
like you said, the different morphologies are fascinating. I mean, even just here along the West Coast, the differences between shallow and deep, and even once you get deeper, they change again, and soft bodied versus spiky versus the armored ones, the squalus that we've been seeing a lot of. So they're quite abundant in a whole bunch of different ones.
Yeah, absolutely. Yeah, I think I didn't see the highlights of it, but apparently there was a herd of sea pigs on a previous dive.
They're always
I
fun.
heard.
Yeah.
They're always entertaining sea pig. Did I say squalus? I meant solus. That's the one that we've been seeing. The white.
Yeah, they're really confusing, right? Because they kind of look branchy like corals, but it's just the respiratory tree they used for gas exchange and capturing particles.
I'm really glad to
it's
have both of
funny
you on the line. I'm more into the policy side of things, and I am not anywhere near as useful as you two are with identifications and talking about the details of species. So I love learning from you both. Thank you.
Everyone is useful on SPL.
Very true.
I'll check.
It is fun to register the disappointment sometimes when you have coral biologists on and see one of those cucumbers in the distance and get right up to it. And, oh, it's a cucumber.
The struggle is real in those. I was last fall. I was doing an Alvin dive with a cruise out of where we're all Southern California Channel Islands, and it's hard when you're looking through the portholes on Alvin to make and distinguish fine features like that. And the disappointment, like you said, is real when you think you found a coral and it's actually just one of those sea cucumbers.
Do those portholes,
like
are they
we have
really distortion-y and thick?
Yeah, right. Typically corals are pretty clean. They're not super well, this is the distinguishing features that I take from it. The corals themselves are not particularly fouled unless they have tissue loss or something that hydroids or some other fauna starts to grow on. And then the same with these trees is that the sea cucumbers keep them clean. They can basically brush them off, eat whatever particles they capture, and then put them back out there. So they both look similar, whereas if it was a sponge or something, you might expect it to be more fouled or full of sediment or debris, as they are sometimes. And they just have this like weird pearlescent color, which is not common in the deep ocean. Usually things are pretty dark and murky.
Yeah, red or black are the two consistent colors.
Do you know why they have that color red?
Looks like
It's because the attenuation
I
of
do,
light.
but
Yeah, ooh, no.
I do, but maybe you should tell us.
No, yeah, so the red color is the first to be kind of removed or attenuated from like going through the water column. So it's kind of acts as pseudo camouflage. To have that coloration is useful.
Yeah, the adaptations to the deep sea go on and on and on. The bioluminescence and the red coloration but just the things that fish and invertebrates are able to do to adjust to living in such a harsh environment is quite incredible. And every animal and anything down there has some special adaptations to living in a super high pressure, super low oxygen environment.
And as I understand it, some of those adaptations are what make them so useful pharmacologically. It does high pressure, very low temperature, strange to us environments makes for animals with all kinds of things that we don't understand and don't know how to produce ourselves. And that's one of the reasons that I think they call it bioprospectors are looking in the deep sea for new cures that we don't know about yet. The little green sponge, I know we're looking for that. That may have a cure for, I think it's prostate cancer, or at least some sort of help for treating that disease.
Well, it's definitely a region that has not been well explored. So using OET and Nautilus and Herc is, furthering our knowledge with every dive.
Steve, I think
We
you
know
mentioned
more
you were
about
gonna...
the moon.
Yes, we do. I think you mentioned there were going to be some introductions of the people on the ship when they were in position. Is now the time?
Yeah, they're still having a little chat up in the front row. Another couple
Okay, we
minutes.
can wait.
Just doing a quick swap out right now.
Gotcha. And I'd like to thank everybody who's joining us on the science chat. We have Abby Powell, Kristen Little, Liz Clark, Katie Rubel, Melanie Johnson, Tim Burbank. I hope I didn't leave anybody out, but thank you all for being here with us and helping us ID things and decide what to collect and helping us make sure that we see everything that's on the screen.
Abby's looking forward to little green sponges.
Aren't we all? And also a huge thank you. I don't know if she's listening to Jenny Waddell, who is the research coordinator for the Olympic Coast National Marine Sanctuary and will be up next on the watch following ours. And she mentioned that she's going to tell the audience all about more of the social and cultural side of the sanctuary, since she knows more about that than anyone else. So we'll stick to, I guess, I can help a little bit with the policy part. And, Erica and Steve and others are absolute experts in the biology. Let's see what we're seeing. Are there any geologists on the cruise today?
I don't think we have any geology specialists right now.
No, but we do reserve the right to wake up Andrew Thurber if we come across any seep activity.
That's right. He's out and about. He's I just saw him this morning. Yeah,
I'm
it
amazed
was
because he pulled a long shift yesterday.
it was a long day for all the onboard scientists yesterday.
The ROV guys too.
Yeah,
I'm sure.
there's they were troubleshooting some arm issues which they fixed, and everything's working out.
Excellent. Herc can
That's
scratch
great.
his back now.
Yeah, that's right.
Fabulous. So I know a little bit about seeps. I studied them a little bit in grad school, but what are we looking for there? Bacterial mats, bubbles if we're lucky? What should we be paying attention to?
Yeah, that's what has been the protocol. So actually we've found some seeps in our multi-beam data in the water column. If there is bubbles, we get to see that. And then on the seafloor, it seemed like the diagnostics were mostly looking at those big microbial mats.
They've been taking a lot of core samples, which Steve mentioned, and trying to get a gradient as you move away from the seep to see how the sediment changes.
Yeah.
He's been collecting the actual bacterial mat itself to determine which bacteria are making the mat. They've been collecting carbonate rock to see what's making up the rock and what's actually growing on it. And they've been collecting several different species that live near the seeps, clams, urchins. I watched him grab some sponges and corals yesterday. So just about anything associated with a seep area is of interest.
Yeah, it was neat on that dive yesterday that as he was doing the gradient across the microbial mat, the seep site, right around the perimeter was this big polychaete matrix, these worms. So that was kind of interesting to see.
Oh, I missed that.
Yeah.
when I was in grad school, I studied the sea urchins that were living near the Santa Barbara oil seeps because they didn't seem to mind one bit being right next to the seep environment. And so studying the transport proteins that they have on the outsides of their cells that may or may not let in the toxicity that's coming from the oil seeping all around them. It was a pretty fascinating project. Of course, it led to more questions and answers, and that some of the urchins' proteins were kind of acclimated to living at a seep, and some could become more acclimated.
Next target is going to be, let me zoom out. We're just basically going east up this cliff. This is a slope map, so it's a little confusing. But this will be T, the first one. You see? Okay. Yeah, we're going, I'm going to put us at like, let's see. Probably like 080. Yeah, these slope maps can be annoying, but they're also helpful. Because I know. I really wish the nav-S had more functionality. Oh, they stopped doing it. Yeah.
Hey, folks in the front row, are you set for an introduction? If you have a chance?
Yeah, I think we're all good. Yeah, I'm Ben Gracian. I'm the sitting in the nav seat.
I'm Bob Waters. I'm the Herc pilot.
I think you're muted. I didn't hear that.
My name is Jake Bonney, and I'm in the Argus seat.
And Dave is on video, but he is troubleshooting something right now.
Okay. Very good. Hi, guys. Good morning.
We're about
150 meters.
3 or 4 minutes from the bottom, so any few minutes we'll have the bottom in sight.
Excellent. I'd like to note that Jenny Waddell letting us know when there are things that we should be collecting, and especially exciting things that we've come across. And she'll also be taking over for the next watch shift. I can't wait to see what she has to say about more of the cultural and social side of the sanctuary, since we've given you a little bit of background on some of the other aspects.
Hey, Bob, can I get power to the still cams?
Yep.
Thank you.
So I think this is our third dive in Quinault Canyon, maybe fourth on this cruise, but it's big, and we're visiting different areas each time and seeing different things. So what amazes me about these canyon areas is the amount of mud that can hang on the steep side of a cliff.
I never really thought about hanging mud before. How does that work?
It defies gravity. I'm not sure how it works, but you would think these
That's
cliff
crazy.
walls would be would be rocky and lots of hard substrate for things to grab onto, but there's just so much sediment that it's impressive that anything manages to grab on.
Oh, well, I'm looking forward to seeing that. I'm not sure I've seen hanging mud before. So I think our plan is to, after we arrive on the bottom and check everything out, to make a gradual ascent along the steep flanks of the ridge that we're targeting. And we'll be slowly ascending to the surface of the ridge. And along the way, we'll be starting transects that I think are about 10 to 15 minutes long. And along those transects, we'll be counting after, well, both during and also after the dive, be counting all of the creatures that are within transect boundaries. We can get some sort of estimate of density. Then once we're up on the summit, we'll go along the ridge top and explore habitats on both sides of the ridge top as much as we can.
Oh. At the bottom.
Want to reduce the range?
Yeah.
Reduce the range.
This is our third dive in Quinault for transects and surveys of coral and sponges. And a couple days ago, we did sample a good deal of sponges from one of the sites, and the mud you were talking about is pretty consolidated stuff, so it doesn't surprise me that corals and sponges can attach. But if you were to manipulate it with your hand a little bit, it does, you know, fall apart fairly easily. But it's pretty tough stuff. It's pretty packed. But of course, there's that, you know, the marine snow layer, that soft sediment layer on top that tends to be very lightweight stuff and rinses off pretty easily.
Get hits on your altimeter?
Not yet. It's jumping around still. I mean, I'll decrease the range again.
What's our expected depth?
It should be 1100.
1090, I think.
Well, yeah. Okay. Oh, maybe this wasn't
on the dive.
You all stop?
Yeah, yep. I'll stop.
That's 1112.
Oh, okay.
Erica, would you say your main expertise is in fish or corals or both? You seem to know what pretty much everything is. So do you have a specialty?
I do not have a specialty. I'm more of I know a little bit about a lot of things.
Just hold. Just hold. Get your hand off the stick.
My first job with NOAA, I worked on the groundfish trawl survey for 10 years. And that survey goes Canadian border to Mexican border 2 times every summer, sampling for information to use in stock assessments and just to monitor what species are out there. So we would do a 15-minute trawl and then count and sample everything that came up.
On? Match me.
So it involved knowing a lot of different species, both fish and inverts, and being able to identify what species we were seeing.
Excellent. How long does it take to go from border to border on a cruise?
We start in May and finish up in October.
Wow. That's long time on a ship. My longest cruise has been about 30 days, something like that. Nowhere near as much. So I imagine you learn quite a lot immersed in that world for months and months.
All right, I'm gonna reset you.
You do?
Yep.
You do see a lot, yes. I'm gonna let these guys get settled for a few minutes
Yes.
as we're on bottom, so I'll tell you a little bit more in a little while about how we run the survey.
Sounds great.
Do your white balance there, Dave?
Yes, sir. Thank you. Perfect. Right there. Okay. Eric's going to go to black. And
Good?
we are good. Thank you very much.
Okay. Zoom out.
I'm gonna do one last reset.
Okay. Want to zoom in just a tad there, Dave? Good. Okay.
All right, we all ready
We're
to?
all dialed in.
so we have about 500 meters to this first target, and it's going to be
080?
500.
No. Heading?
It's going to be 080, so I'm going to put that in now. Definitely looks like there's a lot of organic matter, particulates.
Yeah.
Harder to see. Yeah, so we're going slightly upslope, but kind of following the contour more than going up it.